Clements. And that's right. I'm an actual scientist.
Yeah!
But I will not be doing any science tonight. I will be making up everything on the spot, inspired by our lovely scientist guests. Before we bring them out, let's meet the rest of the team. And now, let's pass it to my friend Sam.
Hi everybody, my name is Sam, and while I might not be that kind of scientist, I am a computer scientist.
Woo! Yeah.
No matter what Tom thinks. Folks, we've been arguing a lot backstage about kind of who's the smartest person on our team. And I'm comfortable enough that I know I'm not. But we gotta figure out who amongst our people are. So we're gonna do a game that we like to call classifications. I'm gonna come to you for a suggestion, and they have to list off something in a category of your choosing. So for example, if I came to you and you gave me colors, it might be something like
Red. Blue. Elephant.
okay, elephant is not a color, which means Tom is now out. When somebody is out, I will call them out, and I will say something like Tom is, and then we'll all clap twice and say out of here. Let's practice that. Tom is
Out of here!
beautiful. Now, who's got my first classification ready? Who's got a suggestion for me?
Animals.
Animals.
Penguin. Cats.
Hippopotamus. Aardvark.
Elephant.
Rhino.
Kitty cat. Bunny.
Tiger. Bear.
Human. Parakeet. People. Partridge.
Pages. Out of here! Now I need a new classification category.
Board games.
Board games.
Trouble.
Monopoly.
Sorry. Sorry.
Sorry, here it is. Out of here! Who's got my next classification?
Areas of study.
Areas of study.
Economics.
Science.
Biology. Biochemistry. Developmental biology. Cell and developmental biology. History. Physics. Astrophysics. Marine biology. Equestrian biology. Etymology.
Engineer.
Entomology. Spanish. Portuguese.
Theology.
Brazilian. Jewish studies. Hindu studies. Christian studies.
Yeah.
I'm bored.
Routine is down to our two potential smartest people, the clones. Who's got my final best juicy category?
Scales.
Scales.
Yes.
Bathroom. On a fish. On a reptile. On a shark.
Doctor's office.
On a dinosaur.
Dinosaurs had feathers. Mary Claire wins.
It feels good to win. And as the smartest person in the room, it is my pleasure to introduce you to the actual smartest person in the room, Aiden Houcek. He's a PhD student at Vanderbilt, and he is here to enlighten us all. Please welcome Aiden.
Alrighty. Well, thank you, everyone. So yeah, my name's Aiden. I'm in the same lab actually as Camille Wang, who's on our improv team. So I feel honored to talk about something that I'm actually not an expert in, which is Crispr. And so to start off, just basically what is CRISPR, how does it work, and why is it so sort of revolutionary? And that's, or one of the ideas is that CRISPR is super precise and really good at finding something. And so CRISPR is really made up of two things. So one is this Cas9 protein here, sort of like looks like a cloud. The other is what's called the guide RNA. And the guide RNA is able to be programmed. It's interchangeable. You can design a guide RNA to target any region in the genome. And so there's about 3 billion individual molecules of DNA. And a guide RNA can find exactly where you want to put it. And with that, the Cas9 will make a cut in the genome. And that cut can get rid of a gene, you can cut the DNA and then put in a new gene. And so a lot of this is super, applicable to a lot of fields of study. So I'm actually in a neuroscience lab. We study how neurons communicate with each other, in particular these little points called synapses, where information is transferred. So we use this in that context, and I'll talk about how we do so. So the next slide. So first of all, I'll say we actually do CRISPR in stem cells. So we don't actually do this in neurons. And we make almost identical stem cells. And they only vary, the two stem cells, if you compare all of their DNA by the one change that we have made using CRISPR. And so we like to talk about as scientists in the lab to keep things very, very controlled, right? Everything is the same except for the manipulation that we have done. And so that's what we do. We take these stem cells and then we use CRISPR on these stem cells, change the DNA, and then we turn these cells into neurons. And so I'll just say that this was something that we actually had to start from square one in the lab. We had never done this before, and so it took a lot of reading and looking at the literature to figure out how we were going to do this. And so it was just so crazy that this was months worth of work, and then on the day we had one last experiment to do, right, to determine whether or not this had worked. And on that day there was a few people in the lab that actually came in to take a few pictures of the graduate students doing research and did a few interviews. And they actually caught my reaction to this experiment working. So I don't know if we have my reaction, if we can go to the next one. You can see how surprised I am that this actually worked. And so yeah, the green means it's good. So, so I said that we do this in stem cells. And so why do we do this in stem cells if we're studying neurons, and what makes sort of stem cells so unique? And that's one of the things is that stem cells in a really weird way are like transformers, in that stem cells have first the ability to divide and keep growing as stem cells, but they also have the ability to become any other cell type. So stem cells make your heart cells, right, cardiac cells, they make neurons in our case, they make muscle cells, and basically any other cell in the body that you want to study. And so that's what's really sort of unique about stem cells, and the reason why we do CRISPR in stem cells is because we can sort of preserve and keep growing these stem cells and then turn them into neurons. So again, when that same group was there, they asked me to sit down and talk about how I feel about stem cells, and yeah. Yeah, cheesy. But so to wrap up, so this is kind of what we're doing overall, in that we have these stem cells and we're using CRISPR to change, and in our case, get rid of certain genes. And those genes, I should say, are ones that are implicated in regulating this transmission at synapses, right? We don't really understand what these genes are doing, but we want to figure it out. So we get rid of these genes in stem cells. And then, like I just showed you, stem cells can become any cell type that they're essentially told to, right? And so we, over years, have figured out how to tell a stem cell, to become a neuron. And so we use two different sort of things. One is what's called a lentivirus. And that is basically a harmless virus that we have programmed and put in our own DNA into this virus. And that virus, we put it on these stem cells, and it goes into the DNA of those stem cells and starts changing the DNA so that they become more like neurons. So that's one thing. The second thing are these neural support factors. Basically, the right nutrients and chemicals to help these stem cells sort of differentiate into mature neurons, right? And so this is sort of overall what I do in the lab and others as well, and ideally gets us one step closer to understanding how the brain works. And so with that, thank you. And that's it.
That's really great. Wasn't that so interesting, everyone? So the next game we're going to play is called States of Matter. We're going to be playing with states of matter, solid, liquid, and gas. And so my name is Camille. I forgot to introduce myself. I'm a PhD student in the same lab as Aiden. And so with this game, our performers will be creating a scene while maintaining the 3 states of matter. So however they feel like that represents those states of matter. At any point, the performer can change their state. However, all 3 states must be represented at all times. And so to start us off with, Aiden talked about how stem cells can become other cells. What is a non-neuronal cell that you would like to, if you were to do stem cell research, what would you transform a stem cell into?
Muscle.
Muscle. Your suggestion is muscle. Two.
I'm getting better, Coach. I need a water break.
All right, my patience is wearing thin a little bit. It's been 5 years and you have not gotten any better at push-ups.
I think I'm gonna need another example, Coach.
Of push-ups?
Yeah, 102, 104.
Coach, I think I need to go home.
Oh my gosh, I have a bunch of pansies. Just pansies on this team.
We prefer Girl Scouts.
I just can't take it anymore. It just feels like it's all gonna come back up.
Okay, just let it out. Let out your thoughts.
How are
and
we
the
going to make the state championships in curling if you can't stand and you can't do a push-up?
Well, maybe we could try some positive reinforcement for once, Coach.
Let me think on that. Okay. If it's been 5 years, I can maybe admit that some of this is my fault.
That's so big of you.
5 years of just abs and push-ups, and I don't feel like I'm curling.
That's right. We haven't even tried to curl. Like, is it like, do we curl weights? I don't know what curling is, Coach.
I have failed you. I have failed curling, the sport you signed up for.
That was 5 years ago. We don't remember. You're just every day. You're like, do a push-up. I'm like, all right. And then I failed. I just did one. Coach,
We're getting somewhere. Oh my God. Are you healing? What's happening right now? Hi, I'm Lindsay. I graduated with engineering and I don't use it anymore, so. Anyways, I'm smart too. For our next experiment, we are going to play lab partners, okay? We've got 4 people up on stage, and 2 at a time, they're going to enact a scene. Right here, we have Mary Claire and Sam. They'll create each their own environment. I'll call them to rotate, and we'll return to each pair several times, okay? To get us started, I need a suggestion for Mary Claire and Sam. Anything at all.
Grandma's house.
Grandma's house.
Rotate. And for Sam and Paige, can we get another suggestion?
Disco.
Disco. Rotate. For Paige and Tom?
Long distance driving.
I love this crowd. Rotate. Tom and Mary Claire?
Long distance relationship.
Long distance relationship.
Awesome. Let's rotate and repeat.
Grandma's house.
Grandma's house.
Grandma's house.
Grandma's house.
Yes.
Rotate.
Disco.
Rotate.
Long distance driving.
Rotate.
Long distance relationship.
Rotate. And we'll get started here.
You came back.
Yeah, Grandma. Of course I did.
I didn't think I'd live to see the day.
That makes me sad that you didn't think I was going to come back and visit you.
No, I thought you'd come back. I just thought I wouldn't.
Grandma, you know I hate it when you talk like that. You're gonna live forever.
I sure hope not.
You're always making me go to all these theme parties, okay? Like last week was jungle.
Yeah. I was Tarzan. You were George.
I remember, everybody thought we were the same person. Nobody knew.
I thought that was a cool trick.
Okay, well, can we at least pick a place, like I would like to go to?
Sure, as long as we wear a themed couple's costume.
It always has to be a theme.
It's kind of my thing.
One of these days, Dad, I'm gonna be the best golfer out there.
That's right, kiddo. You can do it.
No!
Ow!
Sorry about that. Look, shape up, kid. Golfing is a mind thing. Okay, it's all about up here.
Call me by my favorite nickname, Tiger. I'm not Eldrick or Kid anymore.
Look, when you earn the name Tiger, I will call you Tiger. Until that day, you'll still be Kid.
Rotate.
Hey, so I was just thinking it would be so cool if you came up here for the weekend. I don't know. I'm just getting nervous. I haven't seen you in like 6 weeks, and it's hard to get in touch. Hello?
Such a long voicemail. Delete. I'll text her back with some like sup.
Sup? Video. I was really hoping that you'd come and visit me.
Oh, that's not a TikTok. Okay. I'll send her back. Well, I'll just heart react that.
Well, I guess that's good. I mean, it's a heart. I can make that mean something, right?
I can't believe it's been 800 years.
Grandma,
You cursed me.
But we're still together, and I don't age.
Now you're taunting me with your beautiful 17-year-old body. Rude.
No, I really thought that looking up that witch doctor and getting this spell so that we could both live forever in the form of that day, I thought that was the best birthday present ever, Grandma.
Well, it wasn't. What was wrong with the mustard party? You were honey, I was Dijon. They're very different.
literally the same thing.
One's spicy, one's sweet.
You're not tasting either of us. So obviously they're not gonna know if we're both turned around.
Well, it's not my fault you wouldn't let people lick us.
That's weird. No.
That's how we sell the costumes.
I don't need to sell the costumes. I don't want. I just.
I get it. No, I get it. You wanted to be yellow, but they were sold out of that costume. Sorry.
Rotate.
Dad, I cheated on my beautiful model wife and she wrecked my car. I want you to call me Tiger.
That's just not gonna happen. You haven't retired yet.
Does it have to be old and frail like you, Earl?
Yeah.
Earn your respect?
You'll probably earn it when I'm dead.
I look forward to death.
Is that a threat?
If you want it to be.
I don't really want it to be a threat on my life.
I want you to call me Tiger in your obituary, Dad.
Fine, Tigger.
So I've circled LAX 3 times, and I'm pretty sure you said that your flight was supposed to be landing. So I'm just wondering what happened to you. Would be great to hear from you. I've already checked all the flights coming in from Nashville, and they have all landed, and I do not see you. So let me know what happened. Thanks.
Sorry, babe. That's just a long email for no one. I'm all about you.
I don't trust you. Alright, so next we are, well, first off, I'm huge. Apparently our group is not very good at introducing themselves. But for our next one, we have a panel of experts, or lab partners, and what we will be doing is we'll bring these 3 people to the stage, and we need an expertise for them to portray. So, can I get anything from the audience? Anything that somebody could be an expert on?
Animal husbandry.
Animal husbandry. And can I get one for Camille?
Animal wifedom.
Oh my God. And for Sam, we need one more.
Beans.
So everybody, okay, so let us meet our experts. We'll start down here.
My name is Greta Equestrian. And I am a Southerner, if you can't tell. I study animal.
Excellent, thank you, Greta. Okay, our second expert.
Hi everyone, I'm Bessie Smith, and I really want to get married. Yep, just want to make someone sandwiches in the kitchen and birth their children. That's it.
Great, thank you, Bessie. And our third?
My name is Baked Ians. I own the biggest bean plantation this side of the Mississippi.
Great, thank you. Thank you so much.
You're welcome.
And so now from you all, does anybody have a burning question that they've been wondering? Like maybe what does time mean? Anything, really. That's a good one, but don't use that one. So can I get a question?
What's a good marriage proposal?
What is a good marriage proposal? Greta?
Well, I'd say probably somewhere in the woods on trail likely on a horse. Because that's romantic and such. That's pretty good, I think.
All right, thank you. Josie?
So I've been thinking about my marriage proposal since I was 5. And I would like to be proposed in Boston at the statue of a George Washington statue. George Washington statue. I would like you reading a book. Specifically, I'd like you reading Tale of Two Cities. And then I would like my future spouse to kneel to me there and ask me if I would marry them.
Baked beans.
Yeah.
Let me tell you about my marriage. I walked up to her and I said, "Hey, Lucille, I got 20 cans of beans in the back, and I'm willing to trade them to your father for your hand." She said no. I upped it to 30. Offer she couldn't refuse.
I know that was a hard one to follow, but do we have another question, please?
Best recipe.
Oh, what is your best recipe? Beth, why don't we start with you this time?
Yeah, sure. So I have a really good cookie recipe. Yeah, it's really good. Chocolate chip, and you mix butter and flour and sugar, and then you add in chocolate chips, and you mix it all together. You mold them into little cookie things, and you cook them in the oven. You take them out, put a little bit of Maldon salt on them, and then you can eat them.
Raise the bacon.
You take a can, pop that top off, grab you a spoon, and you just go to town.
Excellent.
Beans.
Greta.
Well, Betsy, I make some pretty good chocolate chip cookies. Yeah. I make them slow.
To make them fast.
Whatever you want.
Stop reader.
We both got the chocolate chip.
Excellent. Happy for you guys. All right, and for our third and final question, does anybody have one?
What's the deal with Crispr?
What is the deal with CRISPR? Dave, we'd like to start with you on this one.
Crispy beans are pretty good. I got potato chips, and I get ready for bean chips. It's my next big invention. It's going to do a killing. I don't mean murder. My beans do not have poison in them. Don't listen to those rumors.
Thank you. Greta?
Well, I heard that CRISPR can recreate my favorite horse. So I think I'm going to send her DNA to China, because that's apparently the only place that they'll do it now. But Boston, so.
Greta listens. I like the listener.
We'll turn over to Edie. You can finish us up here.
Yeah, shoot. I'm so sorry, and I can't remember what CRISPR was about because all of a sudden I just give out money all the time. So, I love your eyes.
Hey guys, it's me, Sam again. My previous qualifications still stand. I don't care what Tom has told you. Anybody in the room with PhD? Shut up. So then everybody doesn't know this, but at the end of grad school, you have to do what's called a thesis defense. You've been spending your whole time doing this project, and it's very tumultuous and difficult, and now you must defend yourself. Well, we're going to make our improvisers up here do a little bit of a thesis defense right here, right now. The only problem is they didn't write their thesis. So I'm going to get a subject from you. One of them's going to start talking about that. They will have 2 minutes on the clock. At any point, somebody could, in the back line, can clap their hands and shout objection, at which point they can state their objection. I will either sustain or deny it. Then they will take over and talk about whatever they objected about if I liked it. So, can I get a suggestion for somebody to start talking of?
Raccoonology.
Raccoonology. Ready? 2 minutes on the clock, and go.
Raccoons are terrible little animals. They've got little hands and thumbs and they steal things. When I was a child, I had milk delivered to my door. I'm that old. And the raccoons, we had to keep it in a little container. Otherwise, the raccoons would come and rip the top off of the milk and just guzzle it.
Objection. Did you say you had to keep the raccoons in a container? Is that what was said?
I had to keep the milk in a container so the
No, I
raccoons
think you
could
said
have-
I heard we keep raccoons in a container. Let's hear Rach talk about that.
Here's the thing, I don't think we should keep raccoons in containers.
Objection, Mary Claire.
Raccoons can carry diseases, and so sometimes they do need to be in a container.
The audience like them in containers.
Yeah, that's what most containers you think of are plastic with the lid, and there's no air, right? We need to give the raccoons air, okay?
Objection.
We should not give them air because it's more humane to euthanize them if they're being kept in a cage. That's no way to live.
That's more humane. Let's see.
So if you can't keep a creature in optimal living conditions, it's better to not keep them alive.
Objection. There's a lot of people out here. And I'm not gonna kill them.
Yeah, Lindsay's not going to kill a bunch of people. Sure.
I think we need to think about humans in the most positive light. Everyone's trying their best.
Objection. I don't think I am.
Clearly not. Go ahead.
I could try a lot harder. I don't know, last night I slept 7 hours. I could have slept 9. Let me think. This morning I skipped breakfast, could have had
Objection.
Objection, Tom.
Camille, you are enough.
That's true.
Camille needs to stop being so down on herself. Did you know she's getting an MD, PhD? Being a double doctor. That's one more than I have and one more than Steve.
Objection! I'm embarrassed. Let's talk about something else.
Let's talk about how embarrassed you are.
So when I'm embarrassed, like my face kind of flushes. I'm not sure if you can tell. I also had a little bit of...
Objection!
Objection,
When
Tom.
Camille's embarrassed, she listens to Taylor Swift.
The audience doesn't like that. Keep going, Camille. Don't be embarrassed.
So I do like Taylor Swift, but I don't listen to her all the time. What else?
Objection.
Objection, Lindsay.
She's floundering.
Let's talk about that.
I love Camille. She's clearly uncomfortable, so I just thought I would save the day here. Objection.
Objection, Paige.
I think that I should say that because I am actually a superhero.
All right, let's hear about Paige's actual superhero.
Okay, so I am a superhero.
15 seconds.
Objection!
Objection, Tom.
She didn't even say what power she has.
Fly around, power.
Let's hear about how Paige doesn't have powers.
Yeah, so Paige is really nice.
Objection.
Objection, Lindsay.
Paige has a superpower. She's super nice.
Let's talk about that.
Paige is so nice. Like, I really want to be her best friend ever, actually, since I met her. And
5
so I
seconds.
was like,
Objection!
Objection, Tom.
Lindsay's best friends is her 6 cats.
Yeah, that's true.
So Lindsay has 6 cats.
objection.
Objection.
I have 7 cats. So you guys have about 10 minutes. Please go get a drink, come back, get loose, whatever. We're coming back with some more comedy for you.
Understand my intention.
Hi everyone. Thanks for coming back and not leaving. everyone. What a fantastic show, and such hilarious and good-looking people on stage. I am so impressed. It's just a low-key nerd. Like, this is legit. Cool. Well, I am here to talk to you about the realities of cloning. I'm a little bit of an expert in cloning myself. So, but I want this to be a little interactive. So if you have any questions, like, feel free to ask them as we're going over the presentation. So next slide. Too far. So, what do we think about when we think of cloning? Personally, I think of Dolly, the sheep, the first ever sheep cloned in 1996. I also think of Star Wars. And recently, I'm starting to think of pigs. Because we're using a lot of pigs and human cloning to help us with heart, not regeneration, but like making people live longer with pig hearts. And we're going to talk about a little bit of all of that today. So first, let's deep dive into the history. Cool. So, this is how Dolly the sheep was made. So we got an adult. It says Finn, Dorset, Ewe. That's just the type of sheep. Like, it's just a freaking sheep. So then what they did was they took donor cells from a mammary gland of the sheep. So this is adult sheep, an adult mammary gland cell. And then what they did, they grew these cells and they allowed them to starve. When they allowed them to starve, that shuts down the cell cycle. So now these cells, they're not growing, they're not dividing, they're kind of just in like this weird little state where they don't know whether to live or die. So then we jump over all the way to the right, that little sheep with the horns, and this is the donor sheep. So what they do is they remove an egg cell from this sheep, and then using a micropipette, they literally remove the nucleus of the cell. The nucleus is the center of the cell where all the DNA is located. And then now we have that cell without the nucleus, and this is the cool part. Now we can take our mammary gland cell that's been starved and literally fuse it to this cell with an electric shock. So now we have these electrical pulses, and now we have the donor egg cell with the DNA of the original donor sheep. So this DNA has its own nucleus, it completely matches it. So now they started feeding this cell again, and then it started undergoing cell division, and once it reached a certain number of stages, they implanted that into yet a different sheep, a surrogate mother sheep. I believe they worked on about 14 different sheeps were implanted, and only one of them gave birth. And they gave birth to Dolly, as shown here, which is an exact clone of the original sheep right here. And what's cool about Dolly is she was completely normal. She lived a normal sheep life, normal sheep everything, but it was a huge breakthrough for medical science. So when we have huge breakthroughs in medical science, what do? We immediately think of science fiction. So, I think of Star Wars and the Clone Wars. And what's fascinating about this is they're actually not that off in terms of scientific accuracy. All they did here was they took an original DNA from the sample, in their instance, Jango Fett, and I'm still not sure why they picked Jango Fett. Was he like a pure, like was he had the best DNA, or was he just some dude? If there's a nerd out there, let me know. But what they did is they had this original DNA, and then they did the exact same thing as they did with Dolly. They got a donor cell, removed that DNA, and then put his DNA in there, as opposed to having surrogate mothers. They have these nice little tanks you can see at the top, which are what they're known as artificial wombs. And these are actually, scientists were getting close to making these types of things now. But then, this is where they go off the deep end in Star Wars. Some of these clones could be raised to adulthood in a matter of weeks. They range from weeks to months, and according to Wikipedia, which is a very accurate source, when the clones get raised too fast, it leads to mental instability. But when we think of cloning, we need to realize that just because you have the same DNA as someone doesn't mean you're going to be the exact same person. We've seen identical and fraternal twins all the time. They have the same DNA, but they're two completely different people. So in Star Wars, how they do to fix that is they will literally make the clones like just watch TV of Jango Fett's life to like just wrap it in there so they have their memories. And then they also implant memories in there somehow. So that's how they do it in Star Wars. So now, let's go back into reality a little bit. So, this is an actual slide from my genetics class. So you guys are getting what I believe is a very expensive education for a small price of $7 tonight. So y'all are killing it. So let's focus on mice, which is one of the most common animals to undergo cloning processes. So this is a literal picture of a mouse embryo being injected with DNA. Mice are mammals just like us, so this whole process gets very complicated. So what you have to do is you have two mice that will mate with each other, and then they usually sacrifice the female mice and then take the embryos out of her before they even begin to divide. Then they can take these embryos, put it on a pipette, and then you literally inject it with DNA. And then as we can see on the next slide, what scientists are doing, just like Aiden brought up in his first talk, we're doing CRISPR to change their DNA. And then I worked on a lot of CRISPR in grad school in zebrafish, but what scientists are doing right now is we don't really see to like, there's no reason to be like, oh, this is Dolly, or this is Jango Fett, the perfect human, let's try to get it like him. We want to change the DNA to our will. Most of the time, like cure genetic diseases and things like that, but there's probably going to be some evil supervillains out there as well. So Aiden already talked about how we can use CRISPR to knock out DNA. There's two parts, the Cas9 endonuclease and the guide RNA that cut DNA. Then when you cut DNA, DNA hates being cut. DNA cuts, the repair mechanism to fix DNA is absolute garbage, and it usually leads to insertions or deletions, which can completely mess up DNA. We can see that on the next slide. So this is from a project that I did during my PhD. We can see at the very top line, that's a zoomed in version of this sequence of the gene. And that's, we can see all the letters, G, T, A, C, and DNA. And then I had two sequences here where we had a 4 and a 7 base pair deletion. This completely messes up the gene, specifically because both of these are not in multiples of 3. DNA is read in multiples of 3, so when I make deletions that are not multiples of 3, like 4 and 7, this results in a frame shift deletion, and thus the DNA is completely messed up. When you try to make a protein, it's like, oh, and it usually just completely backfires. So now we know this, how we can use CRISPR to modify the DNA in the injected mice embryos, and then, oh, one more, back one way. And then, so because mammals are incredibly complicated, we have to then take these injected embryos and put them back into a different female mice. In mice, they're called as what's known as a receptive female, which means she was mated with a male that was sterile. So we have to make it seem like the mouse is like, okay, I'm going to be pregnant soon, and then we kind of like shove it in there with surgery. So then now we have all these mice that were born, but the wild thing is that they're all mosaic. And what mosaic means is every individual cell can have a completely different set of DNA. So this means that the cells in your feet versus the cells in your hands or your head could have different mutations in there. Like I showed you with the 4 and 7 base pair deletion, you get a 4 here, 7 here, and any numbers of iterations. But as scientists, we want to breed pure transgenic lines that can actually knock it out in the same way. So we have to then breed these mice again and then select for the same mutation. So usually we do what's known as an outcross, where we breed them to wild-type mice to select for heterozygous mice, which means they have one mutant allele, one normal allele. And by allele, I mean a gene copy, because we all have 2 copies of every gene, one from mom and one from dad. Here we go, we're all the science back. So now we can do this, it'll take multiple generations just to get this pure knockout that we all want, and you can start to see how this gets very problematic with humans. We're not going to do this anytime soon in humans. There was one researcher in China who did do this on humans, but it was very problematic. He was very predatory. Honestly, he didn't even tell them exactly what was going on. He kind of lied to about curing HIV. So don't be like him. I want to talk more about how bad he's actually, I think, on house arrest in China, where some people were afraid for his life. But now let's talk about the good stuff. So we can use cloning to save lives. And just this month, we had the first human pig heart transplant. And I know what most of y'all are thinking of. Why pigs? Why not dogs? Other things like mice. So my reason we do pigs is because they're all over the place. They're everywhere. And they're about as big as we are. Like, we couldn't do it with something smaller than us. And what's cool about the pig heart is it's very compatible with humans. Many of you might know someone who's had a pig heart valve replacement, which is very common now. But in order to put a pig heart in a human, we gotta do a lot of things. Mostly because a full-grown pig heart is bigger than ours. So we gotta get the pig at the right stage. And that can take only 6 months of a pig's life, which is so fast. And then we got to do some CRISPR in order to set up all the right things. So this person who got the pig heart transplant, they were 10 genes modified. 4 were modified to prevent rejection of the heart, and then 6 were modified in order for the pig to make sure it can be compatible with humans. So we did things like making sure it doesn't grow, like knocking out things that could like be related to like some pig viruses that we don't want in the human body. And then, fun thing, after I talked about it, the person who received this heart, this was his last chance. So he was in such a bad condition, was in no fit shape to receive a human heart. Previously, he had ignored doctor's instructions of like what he should do after, because he actually has a pig heart valve. So he's like really getting into pig stuff. And this guy was going to die without the pig heart. And what I did find out about, apparently he stabbed and murdered a guy 15 years ago. I know. But I don't want y'all to start being little eugenicists out there, meaning we should do this to criminals. Awesome. So this is just the beginning of this. Like, this is going to become much more common for doctors. And what I like to say is CRISPR is happening. It's not a matter of if, it's a matter of when at this stage. We're not ready for it yet in humans. But when you think about CRISPR hit the stage in 2012. And that's only 10 years, and it's like skyrocketing above it. Within the next 10 to 15 years, like, we're gonna have children, maybe your children or your grandchildren are gonna be CRISPR. It could be in a playground, like, oh, look at that little rich CRISPR kid. And as a scientist who specializes in CRISPR research, I am all for it. Thank you very much. Our fabulous and beautiful improvisers, especially the tall redhead. They look great. Yeah, question.
Which Star Wars movies was Finn Dorset in?
The new series.
Oh, okay.
Yeah, the ones that I personally like, the middle one, and the other ones not so much. Yeah, awesome. So let's bring back those beautiful improvisers.
I'm the one that gave birth. Alright, so I just want you guys to all remember that. Okay?
Were you saying my CRISPR kids aren't good enough?
Yeah,
I'm just saying that when we're talking about evolving human life, I just want you to remember that I gave birth.
well, where's your child? I don't see it. Yeah.
Daycare?
That's a pretty convenient excuse.
Yeah, you've never been around with your kid.
Yeah, you just keep
Or
saying.
stays in the tank.
Yeah, you've seen ours.
Well, it's a lot easier, Frank, when you can contain them in a tank.
I think you're lying to us. Yeah, Show us the baby.
This is like that time that you had a boyfriend from Canada.
Give you this baby?
Maybe. I wanted it to speak French. Ça c'est Simon?
French is not genetic. Although we did CRISPR my baby with no French from the get-go.
Mommy, I got all my Transformers.
I see that. I see that all those Transformers.
This one is gonna be a neuron. And this one's gonna be a muscle. This one's gonna be the heart.
Okay. Cool. You're a nerd.
And you know what that makes you, Mommy?
Is that your excitement, Gideon?
That means you're nerd mom.
Oh, yay.
It's gonna be great. When I become president, I'll be like, I'm so smart, and there's my mom.
Okay. President Mom? Is that your excitement? Huh? Okay.
I just want to be a normal sheep.
Yeah, well, I just want to be a normal sheep too, but I stand on two legs. Science is fucked, man.
So I don't have any hair.
I know, that's like the whole thing of a sheep, is we got wool.
I mean, no farmer wants to buy me.
No one wants to look at me, they see me and they go, what the fuck is that? Have you ever seen a sheep on two legs before?
Yeah, that's pretty ridiculous. Well, why don't we defy conventions? I mean, if we're not normal sheep anyways, why don't we have a family? Start our lives.
We'll cut to our family.
So glad you learned to walk on two legs. Yeah. Look at them, so strong and learning to walk.
What the fuck is that?
Hey, fuck you, this is our kids.
They're beautiful.
I'm trying to raise a flock of sheep here.
And call us by our names.
Marty and Samantha.
Tommy and Danny. And Tim.
Why do we have to stand on each other?
It killed me. It killed me.
We don't like that kind of attitude around here.
We raise you to be strong. One day, you two are gonna drive.
Gotta be honest. You're not very receptive.
Yeah. Yeah. I'm starving.
I keep giving you suggestions. I'm like, hey, we could go down the block, get some lasagna, and you're like,
I'm not starving for food. I'm starving for affection.
I'm uncomfortable with that statement.
And I am immobilized by your lack of affection.
So me not showing you how much you mean to me has made you freeze?
Yes.
I'm sorry, you're gonna be stuck a while.
But I can't do anything. I can't go out and have a normal life. I'm just stuck here in this dark corner.
Look, it's not in my makeup, all right? I'm not gonna come over there and just snuggle you.
Are you saying that affection is not in your DNA?
No. My parents didn't touch me. Their parents.
We can inject affection into your DNA. shoulder. It's gonna make you cuddle.
I hold my beer.
Cut back to the child.
Bye. Do you remember that lab that you're like, hey man, you want a couple extra bucks? Go give your DNA to this lab. And they picked my DNA out of everybody's. Like, I mean, I guess my mom told me I was special, but like, I mean, this is real.
What are they gonna do with your DNA?
Well, they said that they were gonna make like some army or something. Like, that's cool. Like, that's cool, right? Like,
War is very cool.
yeah, pretty like. I think they're like this guy's violent. This guy is like super smart. I just play video games all day.
Cut to her army. I am so excited.
I'm the greatest detective here. I'm very precise. You're stepping on my turf.
Oh, that is the body take. Good body. Well, I'm just saying, I have a card that says best detective.
My card says my name, Crispr. No.
All right, Crisp.
No, Chris.
Oh. All right, Crisp.
Who is named Crisp?
You now. So let's see it. Let's see this best detective shtick.
Oh, what is this? You're just trying to make me do it. Yo! Hey! What the hell, dude?
What's wrong, dude? I just came from the McDonald's.
Important detective
Hold
work.
on, I'm detecting something. The perpetrator always comes back to the scene of the crime. I'm kind of hungry.
This is why you're a terrible detective.
I solved the case. I just gotta go find her.
That is completely arbitrary. Just a
This is
person
not a-
walking through.
It's common CSI tactics. If they come back to the body, they did it. I've seen the movies and the shows.
You came back. What?
Hey, babe. Hey.
Yeah. Hey, oh!
Sorry.
No, Crispr, detective. You
Hey,
cannot.
can I get a sip?
Burger King. Yeah.
Thank you.
All of you, you're all part of this. I've solved it. You.
Damn, you are a good detective.
I don't know. Sorry, dude.
Are you gonna arrest me or?
I'm just a detective. I don't have that kind of.
I'm really scared about this insemination.
Don't worry, it will go very well.
Your accent frightens me.
Be glad that it is not Chinese.
Tie-dye?
Tie-dye, that's what I meant.
Well, no, I mean, that's definitely science pants.
Is she taking a picture of us?
Wait.
She
Oh,
said-
right now? Just test shots. You're good. As you were. As you
Wait,
were.
I'm having a birthday. No,
Put some
Amy,
science in it.
Can you check my teeth?
Oh. Yeah, you're good. You're good.
Okay.
Okay.
Is that what you're gonna have?
What's wrong with what I'm wearing?
Oh, yes, please bring me the powerful podium. All right, I'm Mary Claire, and my science skills are lacking, but my sexy thrills are packing. So for our last bit here, our final performance of the night, we're going to be sharing with you guys our own spicy preferences in Science is Sexy. So we are going to compare our lovers to your suggestions inspired by anything you saw tonight, proving that science is sexy. So for instance, if you said flowers, I would say, I like my lovers like I like my flowers. They bloom when they're near me. Yeah? Science is sexy. So let me get a suggestion from you guys to kick us off. Anything at all?
Pig
Hello. What? Say that again?
Pig hearts.
Pig hearts is what I heard.
I like my lovers like my pig hearts. They're just a little bit bigger.
I like my lovers like I like my pig hearts. I genetically modify them so they stop growing.
I like my lovers like I like my pig hearts. I don't have one.
I like my lovers like I like my pig hearts. They squeal if you squeeze them real hard.
Can we get a suggestion, please? Presidents.
I like my lovers like I like my presidents. They take charge.
I like my lovers like my presidents. Big on inflation.
I like my lovers like I like my presidents, only around for 4 years.
I like my lovers like I like my presidents. 43 white dudes, then 1 black guy.
I like my lovers like I like my presidents. I did not have sexual relations with that
Can you get a suggestion of something else that you like saw on the show and
Clones.
Clones.
I like my lovers like I like my clones. They look just like me because I'm a fucking narcissist.
I like my lovers like I like my clones. Double the pleasure, double the fun.
Get a suggestion of something. Anything at all?
Dolly Parton.
Dolly Parton.
She was in there.
I like my lovers like I like my Dolly Parton. When I go with them, I go, dear Dolly, Miss Golly, oh my God.
I like my Dolly, only 95 days.
I like my lovers like I like my Dolly. They donate millions of dollars for vaccine research.
I like my lovers like I like my Dolly. It's a sex doll I keep in my closet. Stick around for United States of Improv after this. We have another show coming up in February 25th. Don't say 5. February 25th. It's on a Friday at 9 p.m. Hope to see you there. Thank you all. Have a great night.